All right, let's dive right into this explainer. Today, we're meeting a literal microscopic task force that's using biology to solve some of our most complex, human-made environmental disasters. We are exploring the fascinating science of bioremediation.
So picture this, a massive oil spill. How do we actually clean it up without accidentally making an even bigger mess? Usually when industrial waste, pesticides, or crude oil spill into an ecosystem, our first instinct is to roll in heavy, industrial machinery to try and scoop it up. But what if the answer isn't more metal and gears? What if it's nature's very own elite cleanup crew? Literally, deploying living organisms to eat the pollution. It sounds like sci-fi, right? But it's very real.
Here is our mission briefing for today. We'll start with nature's ultimate cleanup crew, then peek inside the remediation toolkit. From there, we'll cover tactics and field operations, look at some real-world pollution battles, discuss the limits of bioremediation, and finally wrap up with the future of eco-biotech.
Okay, Section 1. Nature's ultimate cleanup crew.
So what exactly is bioremediation? At its core, it's an incredibly elegant, eco-friendly process. We're essentially deploying living agents, think microorganisms, plants, and fungi, to actively seek out and neutralize harmful pollutants. And the absolute genius of it? They aren't just moving the toxic waste from point A to point B. They literally use it as a source of energy and nutrients. They feed on the contaminants and convert them into totally harmless byproducts, like carbon dioxide, water, and biomass. It's a completely natural process that we've figured out how to harness and supercharge. But, just like any elite biological unit, you can't just drop these organisms into a toxic wasteland and expect overnight miracles. They need highly specific, optimal conditions to thrive. I'm talking about ensuring we have the exact right microorganisms for the job, alongside a steady oxygen supply and available nutrients. The environment also needs a favorable temperature and pH balance. Plus, the concentration of the pollution really matters. If the toxicity is just too high or the environment is too acidic, our microscopic crews simply won't be able to metabolize those contaminants.
Moving on to Section 2. Inside the remediation toolkit.
When we're actually out in the field deploying these agents, we generally face two main logistical choices. First, there's in situ bioremediation. This means treating the problem right there on site. It's highly cost-effective and keeps ecological disturbance to an absolute minimum, which makes it absolutely perfect for treating massive, sprawling areas like contaminated groundwater. On the flip side, we have ex situ. This involves physically excavating the contaminated soil or pumping out the water to treat it somewhere else, like in specialized bioreactors, land farming, or massive composting systems. Sure, it's a bit more disruptive to move the material, but the trade-off is that it gives us total control over the environment and drastically speeds up the treatment time.
Now let's meet the specific recruits in our toolkit. It's kind of like assembling a specialized heist crew. For microbial bioremediation, we use bacteria and fungi like Pseudomonas or Bacillus, which are incredibly effective at targeting crude oil, diesel, and pesticides. Then for heavy metals like lead and mercury, we use phytoremediation. This is where we deploy specific plants to act like giant earthbound sponges. If we're dealing with really complex industrial organics, microremediation is our go-to, relying on the unique digestive power of fungi. And finally, rhizoremediation. This one is a brilliant team-up between plant roots and soil microbes working together to cleanse contaminated soil.
That brings us to Section 3, Tactics and Field Operations.
So, how exactly does this biological dismantling happen? It's actually a fascinating multi-step chemical journey. Usually it ticks off with biodegradation, where our microbes start hacking those heavy contaminants down into simpler, smaller compounds. From there, some organisms use bioaccumulation, literally absorbing and safely locking the pollutants away inside their own cellular tissues. Next comes biotransformation, which is a neat trick where the pollutant is chemically altered into a far less toxic form. And finally, the holy grail of the whole process, mineralization. This is the complete, absolute degradation of a dangerous chemical until there is literally nothing left but inorganic carbon dioxide and water. Poof, gone.
All right, Section 4, Real-World Pollution Battles.
You might be wondering, where is this actually happening? Well, these biological agents are being deployed all across the globe every single day. Take a massive marine oil spill, for example. We literally deploy huge communities of microbes to go out and gorge on the petroleum hydrocarbons. Right in our own backyards, they're the absolute backbone of municipal wastewater treatment, quietly removing pathogens and organic waste. We use them to bring dead agricultural soils back to life after heavy pesticide use, to pull dangerous heavy metals like cadmium and arsenic out of old mining sites, and to neutralize some seriously toxic effluents, solvents, and dyes from industrial manufacturing. It's just a wildly versatile approach.
Next up is Section 5, the limits of bioremediation. Now, as amazing as all this sounds, we have to stay grounded and look at the limitations. Because let's be real, this isn't magic. Yes, the advantages are massive, it's incredibly cost-effective, hyper-sustainable, keeps ecosystem disturbance to a minimum, and actually destroys pollutants instead of just burying them. But bioremediation can be painfully slow, especially when you compare it to harsh industrial chemical scrubbing. It is also entirely dependent on maintaining those perfect Goldilocks environmental conditions we talked about earlier. It's not a silver bullet for every single type of pollutant either. And because we are managing a living, breathing ecosystem, it demands continuous, rigorous monitoring to make sure the process is actually working. Which leads us perfectly into Section 6, the future of ecobiotech.
This is where things get genuinely futuristic. Scientists aren't just relying on what nature provides straight out of the box anymore. They're essentially giving our microscopic recruits some serious high-tech upgrades. Through genetic engineering, we are radically boosting their natural pollutant-degrading capabilities. We're talking about using genome editing tools like CRISPR-Cas9 to literally tweak their DNA for peak performance. By leveraging bioinformatics, researchers can map out the exact metabolic pathways a microbe uses to eat a chemical. And with synthetic biology, we can custom design and engineer brand new microbes from the ground up, built specifically for complex environmental missions that natural microbes simply couldn't handle.
In the trajectory of this tech, it is moving incredibly fast. Right now, in the present day, we are heavily utilizing that bioinformatics data to map out natural degradation pathways. Looking toward the near future, we're getting ready to deploy smart biosensors right alongside our engineered microbes to provide active, real-time pollution monitoring. And long-term? The ultimate goal is to execute massive, large-scale ecosystem restoration projects, assisted by cutting-edge nanobiotechnology. We're talking planetary-scale healing.
Which leaves us with a pretty profound question to wrap up this explainer. We've seen how powerful natural bioremediation already is. But as we continue to merge raw biology with advanced tech like CRISPR and synthetic design, how far can we actually push this? Will genetically engineered microorganisms become the ultimate saviors of our ecosystems, finally allowing us to undo centuries of industrial damage? It's an absolutely thrilling possibility. And it proves that nature's cleanup crew might just be the most important key to our sustainable future. Thanks so much for joining me on this explainer. I hope it sparked your curiosity as much as it did mine.